Thank you so much for having me. My name is Mallory. I'm a fifth-year PhD student in Russ Altman's group, and we've been collaborating with Chris and the rest of the deep dive team in order to build applications related to drug repurposing, and really our lab cares about drugs in general, so you can see this going in many different ways. And one of the things that I've been concerned with is how you can take literature-derived information, literature-derived data, combine it in interesting ways in order to predict new relationships to drugs. So what I'm going to be talking to you about today is the application base of what's driving us to extract these relationships and how you might build these types of applications. So in the last 10 years, approval of new drugs by the FDA has remained fairly constant. We expected that in 2003, we have the human genome. There's all of this experimental data coming out from universities across the world. And we expected that we would be able to find new therapeutics for a variety of indications. You'd see an increase in approval of new drugs. However, this hasn't happened. And one of the reasons this hasn't happened is that de novo drug discovery is extremely costly, both in terms of time and money. To bring a drug from finding a potential target for a disease, finding a compound that might impact that target, going through clinical trials, evaluating safety, evaluating that the drug will actually improve quality or reduce symptoms of a disease, and then going through the FDA process, which is an entire thing of and of itself, can take almost like between 10 and 17 years, and on average can cost a company $1 billion. So that is a massive investment from the company side because they want to find these drugs, ultimately make money, important. But it's a massive investment. And to get through this pipeline of discovering a new drug, one that's not already out there in the world, discovering a new drug and then getting it approved by the FDA, roughly 10% of compounds that go through that process will actually make it and be approved. And of those 10%, roughly 20% are actually going to make that money back. So that's a major problem. So now you've invested 10 to 17 years, you've invested so much money, and most of those aren't going to actually make enough revenue to offset those development costs. And so what people start focusing on now is the idea of drug repurposing or drug repositioning. The idea here is instead of going to doing de novo drug discovery, finding a new chemical compound, making sure it's safe, making sure it treats a disease, and then going through the FDA, the idea here is that you're now going to find a drug that's already approved by the FDA. In certain patient populations, certain contexts, the drug is safe. And you're going to try to find a new use for that drug. And so really, you just need to find a compound, license that compound from a company, and then prove it works. And then do the FDA process, which unavoidable and painful. But it's important now to say, how can we move from a de novo drug discovery model and do drug repurposing instead and bring more drugs to market, or at least more drugs with different indications to market? And this has been successful. We've repurposed, not we, but everyone out there that's not me, has repurposed a variety of drugs for new indications. And some of them, say, a drug initially indicated for depression, using it now for, say, fibromyalgia syndrome. That's good. People with fibromyalgia would probably like a drug for that. But more importantly, we found cases where you have an osteoarthritis drug or a rheumatoid arthritis drug. And now we found that, one, it's already safe in patient populations, but it can be used in the treatment of a variety of cancers. And so if we can find these drugs that either are treating rare diseases or treating complex diseases where de novo drug discovery is going to be particularly difficult, this can open up an entire new avenue for treatment for patients. And so I'm not going to go into this too much, of a little bit of background. But so computationally, there's a lot of work going on in drug repurposing, where you focus on experimental data, gene expression data, and others, as well as chemical structure-based and protein structure-based methods to predict whether or not you can use a drug for some other disease or some other indication. But what people have realized is that the literature provides kind of the largest known set of all information. And we've been doing experiments in science for hundreds of years. And unfortunately, we're incredibly inefficient in that we write it down. You write it down, you can't really read it in a high throughput manner. you lose that information. People will focus on, the well-studied cases, the cases that are, hot at some given moment, or at least funded by agencies like cancer. But what ends up happening is these methods, these computational methods that focus on experimental data or structure-based data, are now discovering that literature provides this condensed and well-understood overview of the entire field of what we know about. And this is great because what we have currently in biology are these networks of relationship between genes, diseases, and drugs that are curated by humans. And it shows how these genes and other entities within the biomedical domain interact with one another in order to have some effect. So this is just a representation of the cell cycle. Pretty complex if you think about all the genes that are involved. We have similar types of pathways, or you can think about them as networks for the non-biology folks. So you have the relationships between genes and proteins that show the progression of Parkinson's disease, or the etiology behind Parkinson's disease, which is critical for then treatment. A particular interest to my lab, we care about drugs. What we have is PharmGKB, which is the pharmacogenomics knowledge base, a database where we have human curators who sit and read the literature for us, annotate relevant relationships, and also construct these great pathways that show if you take a drug and it goes into a cell, how does it interact with genes or proteins in order to be metabolized? Or how does it interact with genes and proteins in order to have some therapeutic effect? And so these databases are great. I'm a big fan. I'm not going to read hundreds or thousands of documents and annotate these things. We have experts in their respective fields or their respective domains that are willing to do this. And that's great. Hooray. However, literature is growing more and more every year. There's a whole huge backlog of literature. What do you do to keep up with this data? Especially for the paleobiology database, how do you keep up with all of this text? One thing you could do is you can take, say, an entity of interest like a drug or a disease and kind of filter everything else out and try to get as much information at least that's known about that entity and store that in the database. Or you could focus on very specific journals. All of these are a solution, but they're not a great solution because you lose those relationships that maybe aren't as popular. They might be key for understanding how drugs, genes, and diseases interact with one another in order to, say, understand drug repurposing. But if you don't understand what's beyond just those well-known interactions, you're going to lose vital information. So our application goals, first off, was to construct a database of biomedical relationships, what I'm going to largely be talking about today. These include gene-gene, gene-disease, and gene-drug relationships. For any bio folks, I'm using gene and protein interchangeably, particularly because in the literature, authors use them interchangeably. It's annoying. And then on the second part is to combine these relationships in a meaningful way in order to predict new indications for drugs. Now that we have the largest assortment, even with like a very large connected graph that you can prune and filter, of all of known biology, not just well-known biased interactions because people care, people are funded to find those relationships, but the connecting points between pathways, between different biological processes that we're losing currently. So I'm not going to go into too much about deep dive. Chris and other people will talk about it. But what's important here is I'm going to be discussing is on the application side. Because I'm in a unique position where I know about the domain. I don't do experimental data at all because I can't pipette. But in the experimental domain, I understand what's going on in the literature and the domain itself, but also have the computational abilities to actually interact with the system. So I'm kind of taking that perspective today. So as a brief overview, this is with deep dive, not snorkel, though we're currently moving to snorkel now. There are mainly 4 steps if you're thinking about applications. Text, don't worry about it too much. You do it once. You break up a document into sentences, words or tokens, and a relationship between those words. But ultimately what you want to do is define some sort of extractor where you say, what is a relationship that I care about, and what are the features that denote or might describe that relationship? So here you might say a candidate relationship is 2 entities co-occurring in the same sentence. If you see something that looks like that, call it potential relationship. It might be, probably, more than likely not going to be a real relationship, but it could be, and so you want to keep it in the system. You can annotate it with a label. So here you can do a hard label if it's from some external data source with distant supervision, or if you want to do the data programming angle, you can use what's called a labeling function and assign multiple labels that might be very weak, but you might be able to use to increase your predictive power. You then feed in the deep dive, get some probabilities, and at the application user, you're then going to start iterating in order to say, well, what are some sources of error? How can we update the features, update the training examples, update the system in order to improve performance? And so for an example this morning, if you're interested in finding, say, gene-gene relationships or protein-protein interactions, you could say a candidate gene-gene relationship is any co-occurring two genes in the same sentence. You can then apply it to a corpus here. I have 3 PLOS journals. And then what I'm going to kind of very briefly focus on is evaluating against a curated database. Here, I have the database of interacting proteins. This is nice because these are people who spent years just looking at documents and annotating relationships. And oftentimes people talk about, text extraction, text mining, what have you. They want to know how you actually compare against these databases that's already out there. So if you just look at precision, we're fine, you can get up to 76% precision, but recall is going to take a hit when you're comparing against these databases, because what ends up happening is you have these experts that can go outside of what you might say a candidate gene definition is, a gene-gene definition is, in which case, from those 50%, most of them are falling out because we're losing on the gene symbol side. But that's fine. There are also interesting cases where there are relationships that are annotated by the database, but they're not in the document. And so it starts leading you down another path, which you could say is how do you replicate annotators, human annotators who are experts? If you can't really read that document and know that interaction exists, if those entities don't actually exist obviously in the document. We also evaluate against curation, and for a subset, we got between 79 and 83% precision based on sentence and aggregated document level. And again, we have errors that are falling outside of our candidate definition rules. They're based largely on parsing errors. If 2 sentences aren't parsed correctly and you have your entity spanning 2 sentences, that's outside our definition rule. So we have a number of these extractors for both the entities I've mentioned before in the relationship, so drugs, genes, diseases, gene, drug, and gene disease. And on the entity side, we're getting between 80 and upwards of 90% precision. And then on the relation side, we're around the mid-70s. But what's interesting about the relationships is that we're extracting sentences like this one where we say, no, that's not good. Okay, whatever. We're extracting relationships like here where we say, in this study, we have demonstrated a strong correlation between the loss of this gene's protein expression and more advanced stages of breast cancer progression. So instead of seeing a sentence that says, if you have this gene or a variant in this gene, it's going to cause X. Instead, what we see is a more interesting relationship, in my opinion, of how this gene is pivotal, or might be key, maybe not, but might be key in understanding how the cancer progresses, which is very important if you're thinking about cancer, because your treatment might depend, your different treatment might depend which stage of cancer you're in, which cells, if it's localized cancer versus your lymph nodes, or if it's metastasized. It's very important to know which genes are important in advancing cancer progression. For genes and drugs, I particularly like this example of other ones that I've seen. Some of the ones are more obvious, like gene metabolizes drugs, fantastic. But this one's interesting because it says, moreover, we found that patients with this specific gene, exon 23 mutations, could specifically benefit from the drug, although you might need more patients in further studies. What's interesting about this is this isn't annotated in current databases, and it's kind of within that N of 1, N less than 5 category, where there's not that much information about it, not as well studied as, say, warfarin or some anti-cancer drugs, but it might be key for understanding how you could use another drug that maybe is structurally similar to this one, or based on, or this gene, if it's related to an interesting pathway. So in conclusion, we built a system for extracting multiple types of biomedical interactions, and we're actually iterating to improve performance, and we're applying it to a larger database, data set, of nearly 500,000 articles from PubMed Central. And what's really interesting is I haven't gone into this at all today, but we're also working on developing additional biomedical relations applications using Snorkel and the FDA, because the FDA, it might not be enable-based, but it's very important. So the FDA, understanding what's going on at the FDA is really interesting because they can either make you or break you if you want to bring a drug, a device, or something else to market, and they are the regulators. Those meetings are sometimes not the most interesting, but they are really interested in how you can take information from text and apply it to a variety of applications that are related to their interests, of which they are incredibly interested and incredibly excited about this work and this collaboration. So I'd like to thank my advisor, Russ Altman, and of course, Chris and the rest of the deep dive team, as well as my funding. Thank you.
Any questions for Emily? We have a microphone there. Right here.
The question actually isn't so much about the data itself, but the business environment that you're operating in. I'm just curious, the pharmaceutical companies I've worked in the past have been kind of tight-lipped about the data or very controlling of the data they have. Have you run into issues of IP issues in terms of how you're ingesting the data of the sources you're getting from in order to come up with the results? How are companies reacting to your research?
So right now, I mean, we largely focus on publicly available data. So you can, if you're looking at drugs or chemical structures, we don't care about, like, from my perspective, I'm in academia. I don't interface with companies, at least right now, in trying to find new compounds and change, going through the FDA and change indications. But right now, at least from academia side, we only work with publicly available data. So we don't deal with any IP issues. Of course, if you, say, discover a new use for a drug, then there's going to have to be licensing and issues going on at Stanford.
Do you want to comment on how you might do what you're doing by combining publicly available data with a company's proprietary data?
Yeah, so we actually have some interesting collaborations going on with Pfizer, where they, of course, have proprietary data, but we can't share it. And there are interesting, so for literature-based data, there's some, I haven't really talked about it today, but what you can do is combine these literature-derived relationships with experimental data in an interesting way. Say if you look at network-based and you're trying to think of maybe edge weights related to experimental data, combine that in interesting ways in order to, say, screen drugs for different potential indications. We do have collaborations going on in that area, though I'm not directly involved currently. But there are interesting ways that you can combine the experimental data that could be proprietary with literature-based relationship. I find extremely exciting. But then there are, of course, going to be issues of what you can and can't share and how you actually work with the company in a fair way.
As many of you in the audience know, we have a lot of experience here at Stanford working with companies' proprietary data, even including data that has PII, and solving the questions of handling secure data and still being able to publish results without compromising that data. Other questions? Yes. You want to set up your laptop? Oh, I'm ready to go.
My question is again about proprietary data. Another way of looking at this is to say this is something that is public good. Mm-hmm. And a lot of data must be with the FDA because of all the testing that has been done in the past, positive and negative. Would the FDA be willing to open up the data vaults to that type of research?
So would the FDA be open to releasing data, or maybe I misunderstood?
No, we pay for the FDA. We are the taxpayer. Yeah. Okay. Yeah. There's a lot of information in there that may save lives. Mm-hmm. So, fine. there's publicly available data and there's proprietary data, and there are companies working with it. That's fine. Hopefully they find new drugs or new applications. But there must be also a wealth of data that is owned by the public in the filing cabinets of the FDA. So can that be used?
So the FDA, one of the issues with the FDA, so one of the things I'm working with the FDA is part of what's called the CERSI collaboration with UCSF, where we're trying to apply deep dive to combine disparate data sources across the FDA that's not easily accessed currently, but is publicly available, and then build predictive models on top of that. There is probably, I don't work with the private data from the FDA currently. However, they do have the open FDA where they are releasing a lot of their publicly available data in an easily accessible format. And after that.
Just to add in some color. So one of the things that's been really exciting about working in the biomedical space is it was a number of years ago actually that the NIH put a funding mandate, in their funding mandate, that you actually had to put your research into the public domain and you had to publish in an open access way. And so that's, they've had knock-on effects where every couple of years they've sort of redefined what open means and being more and more open. And so there's a lot of data out there that's, still in text form, but is massive. The FDA, one of the problems or challenges that they have is that that's company and proprietary data that's inside for various drug trials. And the FDA has a bizarre role in the ecosystem where they will actually somehow communicate between different companies bits of information. So they're sort of an arbiter for that information. Now, in their role as arbiter and seeing all of those things, they will actually give someone like Emily in Russ's lab access to the data, but they won't make it publicly available because that would reveal, say, Pfizer's trade secrets or something like this. So there's an interesting role for this sort of technology to play in a public-private sense. But from the perspective of deep dive, it doesn't matter. I mean, it's installed on public things and very private things, and we're going to talk about medical records next, and that will never be released, but it's something we can run on.
Thank you very much, Emily.
Mm-hmm.